So in this workshop, we're going to be thinking about protein structure. We're going to think about the structures of amino acid molecules, like this one here, through to peptides and the peptide bonds that form, through to complex three-dimensional structures of proteins. And we're going to be using various different protein visualization tools in order to investigate protein structure. Now, some people get a little bit intimidated by protein structure. You don't need protecting from it like this mother meerkat is protecting her baby. But this is actually quite interesting in terms of protein structure, because the reason that there's such a strong bond between that mother meerkat and her baby is due to a hormone called oxytocin, which has become known as the cuddle hormone or the love hormone. And it's a hormone that is produced after childbirth. It's a hormone that's produced when mammals breastfeed. So it's quite important for these really deep attachments. So how does this structure of this oxytocin molecule work? Well, oxytocin is effectively just a very small protein. So it's made up of amino acids. So we start with the amino acid glycine, and then we've got the amino acid leucine, and then we've got proline. And those are all linked together through peptide bonds. So peptide bonds always have this characteristic. It's C, double bond O, and then an N and an H, linking to the next amino acid. So you can see that's the structure we've got there. It's just upside down in my example. So how did we get there? Well, let's start off with one amino acid. So we've got our C alpha carbon in the middle. That is linked to an NH2 group. So that's our amino group. We've then got C to a carboxylic acid group. So C double bond O and an OH. We've got variable R group, and that's different for each amino acid. And then we've got a hydrogen. So if we have a second amino acid coming in here, then there's our amino group. Again, we've got another R group, and we've got another H. The carboxylic acid is off the other side. So what we get is a reaction happening between these two molecules. So what we end up doing is we end up losing that OH, and that OH go to become H2O. So that's why it's called condensation reaction, because we make water and we get a new bond forming. So this bond dissolves here, this bond goes there, and then we get a new bond forming between our carbon and our hydrogen, carbon and nitrogen, which gives us the peptide bond that we had up there. So this is a condensation reaction, and that's how amino acids become linked. So we can see there's a number of different peptide bonds in this oxytocin molecule. So I've highlighted one, then there's another one, there's another one. So you can see all through the molecule, we can pick out these very characteristic peptide bond formations. So if we keep making peptide bonds with a long chain of amino acids, then we eventually get to a complicated structure like this. So this is a structure that's quite similar to the receptor for that oxytocin molecule. So the oxytocin will bind to this protein, but this protein has quite complicated structure in its own right. So you can see we have, so here, for example, that's an example of an alpha helix. So what we've got here is so this is a cartoon representation. So we've got round the backbone, we've got the carbons and the nitrogens, as we were talking about before. So they're going around that backbone there. But they're then bound to each other with hydrogen bonds. So we actually get hydrogen bonds forming in like that to stabilise that helix. And that is an example of secondary structure. But what we've also got is this quite complicated arrangement of the protein overall. So we can see we've actually got multiple alpha helices. So we've got an alpha helix going through there. We've got one going through there. We've got a little one in there. Okay. So overall, that folding is known as the tertiary structure of the protein. So the secondary structure is these sort of small regular bits of structure, and then the tertiary structure is formed by sort of combinations of those. And importantly, and we'll think about this in the tutorial, the secondary structure is all about hydrogen bonds from the backbone, whereas tertiary structure is all about the R group, so the different amino acids that have different properties, and there's a variety of different types of bonds. So we can have ionic bonds, we can have hydrogen bonds, we can have hydrophobic interactions, we can have all sorts of things going on to make up our tertiary structure. And the way that that protein is organised helps to determine its structure. So if I just superimpose where this protein is living, so this is a plasma membrane protein, so our oxytocin molecule would be coming in from the surface, and then that will be binding to something on the outside region of this protein here. Then we've got some bits of the protein that go through the plasma membrane, which, if you remember, is a lipid environment. So we might expect that those areas of the protein were more attracted to lipids than other bits. Then we've got the bits in the cytosol here, which of course is an aqueous environment. There's lots of water molecules there. So we might start to ask questions as to what is different about the amino acids on the protein at that point that's in the membrane versus these amino acids that are there exposed to the cytosol. So the tutorial is going to help us to think about these different layers to understand the bonds inside the proteins. So there are 5 sections to the tutorial that will kind of guide you through that. So the first section will start with some amino acids. So we'll think about 3D molecules like this. We'll then build it up to peptides, thinking a little bit more about the peptide bond. We'll then think about secondary structure, then tertiary structure, and then proteins in their cellular environment. So all the way along, there'll be quizzes for you to have a look at. You'll have 3D models to have a look at. You have to score full marks on each section to move on to the next one, and we do want you to do all 5 sections, but we'll give you feedback on your answers. You can work with each other to help you answer the questions. But hopefully when you've done that, then you'll have a really good understanding of the basics of protein structure.